Go for it.
Okay. It looks like we still have a couple more people joining in. Oh, it looks like I could admit on my end as well. Okay.
Yeah, there's a couple still joining, but.
So one thing we should also check, Kat, is I think once we share, I think we could continue to admit people as we get started, actually, because we already shared screen. So I think we should be good.
I'll keep an eye on the waiting room and continue to admit them as you give your intro.
Oh, great. Thank you so much.
Mm-hmm.
All right. Well, welcome everyone to another semester of BE seminar. We'll have a little bit of a different format this year. These will all be taking place over Zoom, so we won't be, as usual, jam-packed into town 337. But you'll be able to get to enjoy it from the comfort of your home. So I think you'll all enjoy this. Kat was gracious enough to set up the chat box and also the raise hand. So when we have questions throughout the seminars, feel free I'll field the questions. And we have a good question from Christina. Where can we find the recordings? That's a good question. So we'll get you an answer to that as well. But we have a great lineup of speakers throughout this semester. And we'll actually be starting this semester with a good friend of mine, Dr. and soon to be Professor Quinton Smith. I've had the pleasure of knowing Quinton for a while now, Dr. Smith. Ever since I was a faculty candidate at Johns Hopkins years ago, and I met Dr. Smith as a graduate student in Sharon Gareck's lab, who's a good friend of the Jason Burdick lab. And I've never told Dr. Smith this, but one of the things that I appreciated during these faculty interviews are they're very nerve-wracking, the tensions are high. And one of the meetings I'll never forget was meeting Dr. Smith as a grad student and the kindness that he showed me. And it really gave me a lot of confidence throughout my faculty interviews. So Dr. Smith graduated from Johns Hopkins University in chemical and biomedical engineering, won numerous awards, the NIH F31, NSF GRFP. He was a Siebel Scholar, which is awarded to the top grad students in the top bioengineering programs. And then we were reunited again. I went on to, I was still a postdoc at the time at MIT at the Koch Institute, and Dr. Smith and I actually reunited at one of the Koch Institute holiday parties, which took place at Cape Cod, which was actually a Halloween costume party, which is a lot of fun. And now, perhaps, one of his greatest honors that he's won, Dr. Smith won the HHMI Hannah Gray Postdoctoral Fellowship, which is one of the highest recognitions for postdoctoral fellows, and provides him support for not only his postdoctoral fellowship, but also for his new lab. And, I want to congratulate him on here and embarrass him a little. He'll be starting his new lab at UC Irvine in chemical engineering and stem cell institute at UC Irvine starting in 2021. So congratulations, Professor Smith. And Dr. Smith does incredible work at the interface of iPSCs, so iPSC stem cell engineering, biomaterial science, so that we could engineer new vascular tissues, and also regenerate liver tissue as well for tissue engineering applications and also personalized medicine applications. He has a wonderful seminar in store. So welcome, Professor Smith. We're very happy to have you at Penn Engineering. I wish we could be in person having cheesesteaks together after your seminar. Just means we'll have to invite you again after you give a great seminar in the future. So welcome, and we very much look forward to hearing your talk.
Well, thank you so much for the kind introduction. It's really interesting just to be called Dr. Smith. I'm not really used to it. And now Professor Smith, so it's a very exciting opportunity. I'm really excited to present some of my PhD work and kind of avenues into my postdoctoral research under the mentorship of Dr. Sangeeta Bhatia at the Koch Institute, where I really focus on liver tissue engineering applications, whereas my PhD work, which I'll talk about, really focuses on the promise of pluripotent stem cell technology. One really cool thing about working in the liver is that it has this unique ability to regenerate. You can actually remove 2/3 of the liver and it could fully grow back to its original size. So it's a really interesting organ. It really plays a lot of roles kind of in the regenerative medicine space. So this research is really motivated by a very grim statistic. In the United States alone, over 114,000 people are in need of a life-saving surgery. So in the majority of cases, patients are in need of a kidney. But as it pertains to the liver, for example, 12% of these patients need this new organ, and annually over 8,000 people die. So with the kidney, we have like extracorporeal devices such as the dialysis machine, but it gets really difficult in terms of really recapitulating the function of the liver, for example, because it does so many different things. It's actually, it's accounted to do over 500 different functions. So what kind of motivates me is the promise of stem cell technology, where we can take a patient's own cells, we can take their fat tissue, skin, hair, and reprogram them to a pluripotent state where they exhibit these unique abilities to differentiate or mature into any cell type in the body. And we can manufacture and scale up these regenerative tissues to virtually any cell type in the body and reintroduce it to a patient so that those cells can act to augment or replace the function of these diseased tissues. So in my PhD work, I really focused on the vasculature. I think this is really what all tissue engineering hinges upon. The majority of your cells and tissues within your body reside within proximity to 100 to 200 microns away from a vascular tree because of the diffusion limits of oxygen and nutrients. So we really focused on the promise of using stem cells for generating a vasculature system. But there are some challenges when we try to use these amazing regenerative cells. One of the true difficulties are we really don't have a strong insight into how these pluripotent cells translate into actual human developmental events. Another issue when it comes to differentiating cells from pluripotent stem cells is that there's typically poor differentiation efficiency and a lack of really reproducible ways to expand these cells. And that could be due to a variety of reasons, including differences in cell source, the utilization of growth factors or serum, for example. So there's a really big emphasis in the field to really streamline these processes. And once we get the cells that we think they are, we have to create different technologies to really evaluate their function and the degree of maturation once we get these tissues. I would like to pitch or propose that we can use engineering technologies and perspectives to really drive at these really fundamental questions within stem cell biology. So how can we use, for example, biomaterials, the mechanics of an environment, microfabricated technologies to really push our understanding of how cells respond to their extracellular environments. So just as a way to orient the audience about my particular research, I want to highlight this process here called gastrulation. And this actually occurs when the embryo is developing, allowing the specification of three distinct germ layers, namely the ectoderm, mesoderm, and endoderm. And these cells give rise to the different tissues within the body. I would like to highlight the mesoderm, which gives rise to the cardiovascular lineage. What's really important to take away from this slide is that early progenitor cells that give rise to these other cardiovascular lineages first have this marker called brachyury, or T, which gives rise to these FLIC1 or VEGFR2 responsive cells that continue to mature into endothelial cells, for example, skeletal muscle cells, or cardiomyocytes. When it comes to the vasculature, it's actually the first organ system to develop. They actually arise in a process called vasculogenesis. And this is the de novo formation of blood vessels. You'll actually get a vascular plexus that arises 2 weeks post-development. So you almost have this primitive vascular tree that arises within the developing embryo, even before the heart begins to beat. These vessels continue to mature through a process called angiogenesis, where you have sprouts that build off of that existing tree. And these cells continue to mature and remodel, and you begin to see supporting cell types such as smooth muscle cells that wrap around the arteries and veins. And when you get to the smaller capillaries in which gas is exchanged, you have the presence of the specialized stromal cell type called pericytes. And you can imagine the vasculature is an exciting playground for an engineer to look at. You can look at, for example, different oxygen tensions within the vascular tree. So as you imagine, you have oxygenated blood in your arteries, deoxygenated blood in your veins. You can really control different aspects of physical forces, such as shear stress that goes along the endothelial wall. And you can imagine in a disease state, such as in atherosclerosis, you have a stiffening arteries, stiffening veins with perturbed blood flow, which we can actually recapitulate in the lab using different technologies. So how can we actually get vascular cells from these pluripotent stem cells? So there are many different types of approaches. Really early on, people use something called embryo body formation, where you can take stem cells, you can encapsulate them in these agar wells, for example, and to create spheroids. And the stem cells will self-organize and form any different tissue, but it's very difficult to reproduce. To really increase the specificity of these differentiation protocols, people introduce co-culture methods. So they'll use a stromal cell, like a fibroblast population, and it secretes instructive cues to allow the cells to differentiate. In my PhD, we use something called a directed differentiation approach, where we can plate stem cells on a defined extracellular matrix and begin to introduce chemical cues. So within our differentiation scheme, we were able to get a population of these early vascular cells after an intermediate mesodermal step. And by 12 days of differentiation, we were able to get endothelial cells or endothelial-like cells that expressed PCAM or CD31, vascular endothelial cadherin, and also these stromal pericyte cells that were positive for SM22 alpha or PGFR beta. So in the lab, we can grow these pluripotent stem cells that have like a high nuclear to cytoplasmic ratio. Here are these spindle-like cells or these mouse embryonic fibroblasts that actually help maintain the stemness of these populations. And we can differentiate them to these bipotent cells. I think this is like a nice demonstration of the dual populations that we can get using this differentiation protocol, where we have these endothelial cells in red with corresponding stromal cells in green. So in this talk, I want to highlight 3 different engineering technologies that we can use to control the stem cell microenvironment. Namely, the use of micropatterning tools where we can really define extracellular matrix spaces and confine cells into that region. We can use different biomaterial surfaces to change the compliance of a substrate. And we can use microfluidics to recapitulate some of the physiologically relevant forces, in addition to controlling the architectures of tissues. So really for very early on within my PhD work, we wanted to ask a very simple, but I think kind of elegant question of like, how do stem cells actually differentiate or lose their pluripotency? So we turned to micropatterns where we can have ideally identical differentiation conditions, really varying in the micropattern diameter. So we used these micropattern islands that range from 80 to 500 microns in diameter and gave them differentiation cues. And we wanted to see the loss of this green over time, over the course of differentiation. And what's very unique about this process is that we can use a very unanimous population of cells. So here's a marker called TRA181. Through immunofluorescent staining and flow cytometry, we can identify that the majority of the cells have this unanimous expression of this pluripotent marker. And then we can place these cells on these micropatterns with varying geometric configurations and look at the steady loss of pluripotency over time. So we have many different conditions, as I mentioned. We were ranging from 80 to 500 microns in diameter. This is a 140 micron example. And to interrogate how these different roles play, or how these different environments play a role in the differentiation kinetics, I wrote a MATLAB image processing program to quickly annotate these individual differentiation environments. So we can see here, the program's able to identify individual micropattern domains. And then we can identify each individual nuclei that's present on that micropattern domain. And then we could do co-localization studies and we can identify which of those cells are TRA181 positive. So here's an example of the identification and fidelity of the program in identifying these TRA180 cells. And just very early on, we found some very interesting dynamics when we allow the cells to differentiate over time in these controlled geometries. So we found that on the smaller micropattern domains ranging in 80 to 140 microns, after day 1, we found a bimodal distribution of cells that were either completely pluripotent or cells that begun to differentiate, whereas we had a more unimodal distribution when cells were grown under these larger micropattern domains. And we find that we see a steady loss of these pluripotent markers when the cells are grown on these larger micropatterns, but we see a maintenance of the stemness when these cells are grown on these 80 to 140 microns in diameter. In collaboration with Sean Sun in the Mechanical Engineering Department of Johns Hopkins, we did computational modeling. We looked at how cells migrate on these micropatterns, but it became a very important demonstration that these local differentiation cues are important. So for example, in different differentiation techniques, you do different seeding densities, for example. And we can see that the seeding density plays a very important role in differentiation efficiency. Kind of to complicate or look more into developmental events using this micro pattern platform, we wanted to see if local changes in geometry, namely the curvature, the radius of curvature, had an impact on how the cells mature or choose to differentiate. So we're not looking necessarily at the loss of pluripotency in these cases. We're looking at the gain of a early mesodermal marker or brachyry that I mentioned to you early in the talk. So we did something very simple. We cultured these pluripotent stem cells, so we dissociated them from their mouse embryonic feeder layer, and then grew them in the presence or without the presence of something called a ROCK inhibitor, which is shown to reduce cytoskeletal tension on these micropatterns. And then after 48 hours, we fixed these tissues and we wanted to look at how the cells differentiated under these confined geometries. So here's an example of stem cells grown on these micropatterns where they're able to survey the microenvironment. And in the time course of differentiation, 48 hours, we can look at this degree of brachyry or T shown in green. And what we found was very interesting. We found in our control conditions with cells were grown on these micropattern domains that we see an annulus or an enrichment of brachyry positive cells around the periphery of the micropatterns corresponding to an increase in something called PMLC or the phosphorylated myosin light chain, which is indicative of cytoskeletal tension. If we maintain ROCK inhibitor conditions to reduce that tension, we completely lose or ablate this self-organizing capacity. And we did this for a range of micropattern geometries and sizes. And in general, we found that there was a strong correlation to this edge effect on the micropattern domains in this brachyry specification. And like I showed in the previous slide, that this temporal maintenance of a ROCK inhibitor within the media completely loses the capacity for these cells to self-organize into these specific brachyry positive domains. So I mentioned to you that this brachyry marker is an indicative of a cardiovascular lineage specification. So what's really unique with these micropatterns is that we can perturb the microenvironment, either through geometry or curvature, for example, and look at the long-term effects of these differentiation cues along the process of vascular specification. And we did just that. So we looked after 2 days of brachyry expression and maintained the cells and media that will promote them to form these vascular lineages. And we found something very striking, that the cells that had this brachyry annulus continued to undergo the differentiation program towards these early vascular cells. So in red, we can see this beautiful annulus of endothelial cells that continue to differentiate in this spatially restricted fashion, whereas temporary disruption of tension completely prevents the cells to continue to undergo this vascular specification, really making it known that early differentiation cues and timing is really important in downstream lineage specification. So we find that micropatterns provide a platform to test varying differentiation conditions in a very high throughput manner, and I was able to use image processing to quickly annotate these different conditions. And we find that geometric confinement and the resulting cell density can induce brachyry organization in a tension-dependent manner. And we find that these organizational events can lead to downstream lineage restriction during specifically to vascular specification. So in addition to mechanical cues and these geometric configurations during differentiation, we know that chemical cues are very important. And this is a typical fashion that stem cell biologists use in inducing specific lineages. So we know mesoderm 6 right. And people have used a lot of different strategies to effectively differentiate cells into the mesoderm using something called a Wnt agonist, where you can use small molecules to inhibit something called GS3K, which inhibits the destruction of beta-catenin, where it allows the cells to undergo downstream signaling. So in this seminal work, we can see here that in the absence of these Wnt agonists that block GSK3, you get this steady shuttling of beta-catenin into the nucleus, indicative of Wnt signaling. In groups such as from Sean Palachek's group, we're able to demonstrate the utility of using this Wnt agonist during differentiation, where they're able to differentiate endothelial progenitor cells very rapidly. So you can see here a beautiful specification of endothelial cells here in red with VCAT here in staining. But we see these progenitor cells actually lack downstream functionality, which is something that I mentioned in terms of really the efficacy of these protocols. So here the cells at this point, although they have endothelial markers, are unable to really show or produce von Willebrand factor, which is really important in the clotting cascade. So in addition to chemical cues, we know that mechanical cues play a very important role in differentiation outcome. I think this is really fascinating work pioneered by Christopher Chen, Stefano Piccolo's group, that were able to demonstrate that the degree of cell spreading mediated by either confinement or stiffness of the substratum actually drives major differentiation events. Using mesenchymal stem cells as an example, they've shown that when cells are under high tension, cells are able to undergo osteogenic differentiation or bone differentiation, corresponding to an increase in nuclear localization of YAP-TAZ, which is determined to be this rheostat or feedback of the mechanical environment. Whereas cells that are cultured on a softer substrate actually undergo adipogenic differentiation, all under the same chemical cues. So this gave a large indication that mechanical cues are very important in downstream differentiation. So inspired by these events, I was in a chemical engineering lab. We use PDMS, it's like my favorite material, and we're able to tune the compliance or the stiffness of the material by just changing the ratio between the curing agent and the elastomeric base. And we were able to use axial tensile testing and demonstrate that we can create physiologically stiff materials on the order of 1.7 megapascals. You can think of that as like cartilage, and physiologically soft around 3 kilopascals, which you can think of as like brain tissue, very soft. We wanted to see, can pluripotent stem cells actually sense that stiffness? So we we used the spin coat of these PDMS substrates, and we were able to put these on cover slips. And then we were able to look at the shuttling of YAP-TAZ on these different compliance substrates and also correlate the degree of cell spreading. And to our surprise, we were able to see that pluripotent stem cells cultured for just 24 hours actually have a robust response to the underlying substratum. So we see that the degree of cell spreading and the YAP-TAZ localization is dependent on the stiffness of the underlying substratum. Motivated by Christy Anseth's group, that we're able to show the priming of mesenchymal stem cell differentiation is really dependent on when stem cells differentiate on particular stiff materials. We wanted to see if we can prime our mesoderm induction on these PDMS matrices, and we wanted to see if those mesodermal pools had the capacity to differentiate into endothelial cells, and if there was any kind of interaction or if there's any contribution to these early differentiation events, as I showed in the micropattern case, on downstream endothelial specification. So we were able to quantify the degree of YAP localization when cells were grown on these PDMS substrates during the mesoderm induction phase. And we looked at the localization of beta-catenin in this process. And to our surprise, we found that on 3 gigapascal substrates, this is typical within polystyrene dishes, for example, we see this enrichment of cell-to-cell localization or junctional localization of beta-catenin, whereas on these softer substrates, we actually see this beta-catenin accumulation in the cytoplasm, which corresponds to a decrease in YAP localization over time when we look at these substrates. And we also wanted to look at the trajectory or the signature of mesodermal genes during this process, and we compared this to 3 gigapascals. And we find that on our really softer substrates, 3 kilopascals and 1.7 megapascals, we see this enrichment of brachyury, this KDR, MES1, GATA2. These are all markers that are really important in early differentiation behavior. And then we changed these mesodermal pool cells towards EC differentiation and found that the cells that were primed under these softer substrates actually underwent really robust endothelial specification. But in the beginning of these studies, we did this in serum-containing media. So to address the issues that I brought up before in terms of reproducibility, we wanted to see if we can reproduce this in completely serum-free media conditions. So we did a similar 2-part differentiation where we prime the cells on softer substrates. We see here on the 3 gigapascals in the serum-free medium, we see this enrichment of cell-to-cell localized beta-catenin. And we see a readily cytoplasmic localization of beta-catenin. And when we transfer them to EC differentiation conditions, we find that we get a robust response of vascular endothelial specification. So here I'm showing flow cytometry plots. In green are human umbilical vein endothelial cells, which are a mature endothelial cell type. And in red, we're looking at the degree of VCAD-adherent expression within our stem cell derivatives that have been primed under these different differentiation conditions. And what we find is that when cells are primed under this 1.7 megapascal differentiation condition, over repeated differentiations, we find a very robust peak of VCAD expression, showing that we're really pushing differentiation. And when we look at other markers of endothelial cells using this technique, we find that under 3 gigapascals, although they produce VCAD-adherent and other markers of endothelial identity, they have very limited von Willebrand factor expression. However, when we continue to differentiate these cells and prime them under these more compliant PDMS substrates, we see a rapid increase in von Willebrand factor expression after the differentiation. So we find that we can use PDMS as a material to control substrate compliance. And then we can look at the trajectory of differentiation kinetics with qPCR along cells that are being primed on these PDMS substrates. And we see that this mesoderm induction really impacts downstream differentiation. So in the last part of my talk, I want to emphasize how we can use engineering tools to recreate the architecture of specific tissues or introduce physiologically relevant levels of shear stress. So here I'm showing a microfluidic device that we can create in the lab using soft lithography, using PDMS as a molding agent. And we can create these rectangular tubes, almost like a blood vessel, where we can expose endothelial cells to physiologically relevant levels of shear stress. And we know that there are many different facets of endothelial biology that enables these endothelial cells to really sense the mechanics of shear flow during homeostasis and also disease states. So a lot of this work has really been pioneered by Martin Schwartz's group and among others, where they show there's like a VCAD, PCAM, KDR complex that is able to sense the tensile forces that are imposed with shear stress. But I was really interested in another type of mechanosensor that I think is really not studied that much. And I think it's a fascinating organelle, which is called the primary cilia. So these are microtubule extensions that reach outside of the cell. It's almost like a little antenna that senses different changes of the local microenvironment. So using this microfluidic platform, we're able to introduce shear stress. We're able to see one of the hallmarks of endothelial response, which is this alignment. So utilizing image processing program and looking at the aspect ratio of these cells, we're able to look at the change of the orientation of the nuclei in addition to the elongation of cells when they're exposed to shear stress. And we wanted to test different shear responsive endothelial cells in this platform. So we looked at HUVECs and a progenitor cell type called endothelial colony forming cells. And we wanted to see if these cells that we know are able to undergo shear stress contain primary cilia. So primary cilia presentation is tightly in tune with the cell cycle. They actually rapidly nucleate in quiescent cells. And we can see this primary cilia presentation after serum starvation. So we see that these cells are equipped with this ability to form primary cilia. So we did a very simple study. We looked at adding a drug called cytoclasin D, which temporarily disrupts F-actin polymerization. And we see that disruption of F-actin doesn't have an impact on the ability for these cells to nucleate primary cilia, evidenced through this acetyl alpha tubulin. And after temporary reduction of F-actin with cytoclasin D, we're able to expose these cells to shear stress over 24 hours, and we can observe their alignment. However, as in the case with these endothelial colony forming cells, if we disrupt microtubules, which is responsible for the primary cilia presentation with a drug called nicotazole, we see that F-actin is intact, but the cells are not able to elongate and align. So we have a platform where we can test the mechanics of endothelial shear response. So we looked at other developmentally relevant models to see if we can incorporate our stem cell-derived endothelial cells in cilia presentation and really looking at how these cells are able to mechanosense. So I'm really fascinated by this work. This work was done by a French group led by Jackie Goethez, where they're able to track endothelial cells during zebrafish development. This is a FLIC1 or KDR line where we're able to see this vascular development. I think it's very fascinating work. And what they were able to demonstrate is that during development, that these endothelial cells of both arterial and venous identity have this primary cilia presentation. And I want to reiterate that endothelial cells, as they develop, do not actually develop with perfusing blood. So the vascular plexus is laid down, and then the blood begins to flow after a cardiomyocyte contraction. So what they were able to show with these beautiful images is that in green, we have primary cilia. We actually see blood being pumped after the establishment of the vascular plexus. So 24 hours post-fertilization, before there's any heart contraction, they were able to demonstrate this freestanding primary cilia antenna. But after the heart begins to beat 26 hours of post-fertilization, they're able to see rapid influx of calcium through this GCamp reporter. In 28 hours post-fertilization, you continue to see an increase in this intracellular calcium influx, which is really important in downstream maturation. And what they were able to show is quite fascinating. So they're able to show in their silent heart mutant, we actually don't have any contraction from the heart, or in this PKD mutant where the mechanosensory elements of the primary cilia is deleted, or in the case of the GATA1 mutant where there's actually flow of blood but without blood, so this GATA1 mutant, you actually see that the maturation of the vascular loops is decreased. So you need the shear or the viscous forces of the blood, the mechanosensory elements present on the primary cilia, and flow to really induce mature endothelial cells. So you wanted to look at how we can use our stem cell-derived endothelial cells, a population that has never been exposed to these physiological forces. So using our protocol, we found something that was quite striking. So we had our bipotent population of endothelial cells and our pericytes. So I'm showing VCAD expression in green here. And we find that after serum starvation, that only the non-VCAD positive cells, or our pericyte population, actually contain this primary cilia presentation. When we expose these early vascular cells to shear stress within our microfluidic devices, here I'm showing 20 dynes per centimeter squared, which is arterial blood flow, we actually see that our pericyte population, equipped with this primary cilia here, and also shown in green, are able to robustly mechanorespond in this microfluidic environment. However, our endothelial cell populations were undergoing the same degree of cytoskeletal elongation in response to the blood flow. So we thought maybe it has something to do with the kind of contaminating population of the pericytes. So using a process called magnetic activated cell sorting, we're able to take our early vascular cells and enrich for these vascular endothelial cadherin positive cells, or endothelial cells. And we can show here through immunofluorescent staining that we have a robust uniform population of stem cell derived endothelial cells. And then when we expose these cells to the microfluidic conditions using arterial shear stress, we find that these cells have difficulty in elongating and aligning to shear stress, whereas our ciliated pericytes tend to undergo a perpendicular elongation in response to shear stress, which we found was very interesting. So we wanted to expand our pool of stem cell derivatives, so we looked into using other cell sources. So we tried other cell sources in a different differentiation protocol, which is another parameter that can play a large impact on the ability for these cells to functionally mature. So we looked at our BC1 cells that had difficulty elongating to shear stress, and then we added an additional stem cell line called C12. We find that when we differentiate them under these controlled conditions, we get a pretty uniform response. So we get very similar outcome in terms of different early markers of endothelial specification, as well as different markers of arterial or venous specification along the differentiation. And when we isolate these endothelial progenitor, or these endothelial cells from the stem cell pool, we can actually grow them. We see they have von Willebrand factor expression. They maintain CD31 and VCAD expression over many passages. So we looked into the degree of ciliogenesis in these cells that were differentiated in a similar way. And then we reached out to Mervyn Yoder, a close collaborator, where they were able to use these endothelial colony forming cells derived from pluripotent stem cells. And what we find was very interesting is that the other endothelial populations were able to rapidly nucleate these primary cilia. So we wanted to challenge these different stem cell-derived endothelial pools to shear stress using this established microfluidic platform. And what we found is that in our non-ciliated population of cells, even when we shear them for a very long period of time, they're unable to phenotypically align to shear stress. Whereas in our other iPSC-derived endothelial cells, they contain primary ciliary presentation and rapidly elongate and align to shear stress after 24 hours, which was very interesting. We wanted to look at other facets of endothelial response to shear force. So kind of motivated by the developmental events and our known understanding of how endothelial cells sense shear stress, we exposed these cells to a stepwise amount of shear and looked at calcium influx. And we found that our non-ciliated endothelial cells had a very aberrant calcium handling when compared to our ciliated endothelial cells derived from stem cells. In addition to this assay, we use something called a wound healing assay, where we're able to artificially create a scratch on a tissue culture plate, for example, and we can look at the motility or the ability for these cells to migrate in a coordinated fashion in response to these cues. And it's been shown that primary cilia, not only acting as an antenna for mechanoresponsiveness, also can orient the cell in response to wound healing. And we find that our non-ciliated stem cell-derived endothelial population actually had very difficulty in closing the gap, and they actually had undirectional migration, so they didn't have a persistent migration to heal this wound. So we looked into what could mediate the ciliated phenotype, and we found that HDAC6 is a strong mediator of ciliogenesis within cells. It controls the degree of acetylation. So we found a molecule called tubistatin A, and we're able to demonstrate that with the addition of tubistatin A, we're able to recover the ciliated phenotype of these non-ciliated stem cell-derived endothelial cells. And we find that in our control conditions, without the addition of tubistatin, we see very high levels of HDAC6 and low levels of acetylated alpha-tubulin, which I demonstrated to be this marker of ciliogenesis. But in the presence of tubistatin A, we actually can recover the ciliated phenotype. So we wanted to look at downstream functionality of these stem cell-derived endothelial cells in the assays that I described to you before. So we were able to demonstrate that with the addition of tubistatin A, we can actually improve the directional migration of these stem cell-derived endothelial cells that were once non-ciliated, and that we can see some degree of endothelial alignment after this tubistatin pre-treatment with our formerly endothelial cells that didn't have the ability to sense the mechanical shear that was present. So in this portion of the talk, I want to highlight the use of microfluidics as a way to really interrogate the functionality of stem cell derivatives. We were able to investigate a naive population of endothelial cells and introduce developmentally relevant cues. And we're able to find that there's possibly the role of primary cilia presentation during early developmental events, and that primary cilia could be really important marker for the degree of functionality of these stem cell derivatives. In my postdoc, I'm really focused on liver regeneration, and I'm really focused on building a vessel-like architecture that's somewhat similar to the blood vessels that I described to you in the first part of my talk. So as I mentioned to you before, the liver does so many different functions, over 500. And one of those functions is the production of bile. So bile is a byproduct of heme metabolism or blood metabolism, and it plays a very important role in digestion. Bile is very toxic, but it acts as like an emulsifying agent. And we find that bile, which is secreted by hepatocytes, or the main functioning cell type in the liver, is transported by this biliary tree. So in the Bottia lab, we also use engineering approaches to really control cell function and cell fate. So in this particular example, I'm demonstrating really the power of using these micropattern technologies. So here we're able to take cadaveric or primary human hepatocytes, and we micropattern them on these collagen islands, and then we grow them in the presence of fibroblasts. So we have a 2-cell population. And we find that both homotypic interactions mediated by cell-to-cell contacts of hepatocytes and heterotypic interactions mediated by fibroblasts play a really important role in maintaining those 500 different functions. So here is a functional readout of hepatocytes within these different culture modalities. And we find that when hepatocytes are just cultured alone, that they actually rapidly lose their function. But when they're cultured in this micropattern configuration, we find that they're able to produce albumin, one of their functional markers. In addition to albumin synthesis, they're able to handle nitrogen metabolism, evidenced through urea synthesis. And in a very similar manner, when hepatocytes are cultured alone, without the addition of these structural cues, the hepatocytes rapidly lose their function. Kelly Stevens, a former postdoc in our group, and now a professor at the University of Washington, kind of took this to the next level, looking at the degree of endothelial incorporation in very defined geometric orientation, where she was able to show that you can stabilize primary hepatocytes in a kind of a 3D format with fibroblasts, and that the degree of interactions with endothelial cells plays a very important role in how these cells function. So in this patterning technique that she utilized, she demonstrated that if hepatocytes are grown in a juxtaposed condition with endothelial cells, shown here in green, that they actually have an abrogated albumin function. However, when you grow them in a paracrine manner, we actually see that the total amount of albumin actually increases, demonstrating that paracrine cues and architecture is really important in maintaining the stability of hepatocytes. Leveraging these tools, she was able to demonstrate how we can use this towards liver regeneration. So she was able to demonstrate that we can stabilize primary human hepatocytes in these agarel formats, and that we can use the fibroblasts as a way to maintain their phenotype, and then introduce endothelial cords in a very controlled and spatially oriented manner, and use this as a miniature liver in challenging it towards a regenerative model. So we use something called an FNRG mouse, which is a model of something called hereditary tyrosinemia, which is a metabolic disorder. And we actually found that with this metabolic disorder, the only really way to treat this disease is with a drug called NTBC. It blocks a toxic intermediate. So what she was able to show is that we can actually use these architectural cues that maintain hepatocyte stability and implant them in an ectopic space. And then harnessing this FNRG tyrosinemia model, we could cycle degrees of injury using this drug as a way to see if these miniature engineered liver tissues can actually regenerate in response to injury stimulus. And what she found was very fascinating. So she found that in the absence of NTBC, in the absence of NTBC, that there are no really regenerative cues, so you don't actually cycle under this injury stimulus. But when you cycle with NTBC, you actually get rapid proliferation of these engrafted hepatocytes. And what she found, which was very interesting, is that some portion of that graft contained biliary cells. So here's a highlight of these biliary epithelial cells that I mentioned to you before, which provides the drainage system from hepatocyte-secreted bile acid. So really taking all this together, I wanted to see if I can engineer another vascular architecture, but namely with the biliary system, to incorporate in these engineered grafts. So this is some general characterization of these biliary epithelial cells. So we can see here that they have this marker CK7. These epithelial cells that are found in the liver are highly ciliated. And then when we culture them in 3D environments, they form this native cystic architecture. But we really want to get these cells to form branch tubular-like structures. So I'm really working at looking at different ways or different modalities in culturing these cells to facilitate the self-organization into a branch biliary tree. So I find that if we culture these stromal cells, these fibroblasts, which I've been really emphasizing as an important instructive cue that's been shown to maintain hepatocyte stability, really what's the role of these fibroblasts in the presence of instructive 3D matrices in really driving morphogenesis? And I find that in the presence of these fibroblast pools, these biliary epithelial cells undergo rapid tubulogenesis as opposed to the cystic formation that they form in the absence of these paracrine signals from fibroblasts. So I think this is really great research where we can really disentangle the contributions of stromal cell populations and really inducing a morphogenic response in these biliary epithelium. But as an engineer and someone who's really, really fascinated with microfluidic systems, I'm really excited that I have the opportunity to work with Christopher Chen. His group is at BU and they do a lot of vascular tissue engineering work. And utilizing these systems, we're able to fabricate on-demand vessel-like architectures that I want to translate to the biliary tree. So we're able to use something that we call soft microfluidics, where we actually use acupuncture needles to create these predefined geometries that act as sacrificial elements. So within our microfluidic system, we can use these acupuncture needles as a mold, and we can use any polymer solution that our heart desires. We can use synthetic or natural biomaterial scaffolds, and we can cross-link these biomaterials across the mold, and then we can remove these needles, which leaves us with an instant perfusible tube. So I'm really interested in trying to induce a self-organization of a biliary tree using these microfluidic systems. So here's an example of how me introducing biliary epithelial cells within these prefabricated 300 micron diameter channels. That really gives us a lot of room for really organizing different architectures within the liver. So here I think this shows the fidelity of using this type of system to engineer these biliary ducts. So here I think is just like a fascinating image where we're able to use this micromolding technique as a way to rapidly fabricate really align and structurally relevant systems within a 3D biomimetic system, as opposed to the microfluidic system that I use in my PhD, where we use glass and a PDMS substrate. In this case, we're able to use a biomimetic material, namely these are collagen gels. It's a lot softer and we can really get to the physiological relevance of creating these architectures in 3D. So in conclusion, I hope I was able to demonstrate or convince to you that there are many different engineering techniques that can really allow us to probe into new insights into new realms of biology, specifically towards how these factors play a role in stem cell differentiation and how we can leverage them to really appreciate or to evaluate the functionality of the stem cells that we can differentiate. And moving forward in my lab, at UC Irvine, I'm really interested in pushing the boundaries of some of these micropatterning tools as they pertain to the self-organization of some of those gastrulate systems. So I'm really interested to see if we can spatially resolve what's going on with many of these cells as they differentiate on these micropattern domains. And we can use spatial transcriptomics, for example, as a way to provide a landscape of what's going on in these differentiated tissues. The liver is very unique in that the hepatocytes are the main functioning liver, and the cholangiocytes come from an endodermal precursor called hepatoblast. So almost like the bipotent early vascular cells that I presented to you here, that the liver has a very similar bipotent capacity. So I'm really interested in teasing out how mechanical cues can dictate whether these hepatoblasts are privileged to undergo cholangiocyte or hepatocyte specification. And finally, I'm really interested in using microfluidic organoid models. So here's just a brief snippet of some of the other works that I'm doing as a way where we can template endothelial cells within these microfluidic systems. We can incorporate these spheroids of hepatocytes and also incorporate biliary organoids within these systems where we can have a microfluidic environment where we basically have a liver on a chip with all primary human cells encapsulated within these devices. And with that, I would like to thank all of you for attending my seminar. And I really want to thank the support that I've had from my PhD work and my postdoctoral research. I'm really blessed to have the opportunity to work with Sharon Direct at Johns Hopkins. I feel really blessed to work with Sangeeta and Chris Chen. Sangeeta is very translationally focused, and it's really opened my eyes of like, what do we think about when we try to build these tissues, and what's the importance of scale up, or how can we use this to actually treat patients with liver disease? And it's just been a really awesome time working with Chris Chen. Their work is really fascinating. It's great to see how you can use very elegant engineering tools to start decoupling some of the intricacies of biology. And finally, I really want to thank HHMI. I think the support that they've given me for really kickstarting my lab is something that's been amazing. And I really thank HHMI for their support. So with that, I'd love to take any questions.
Thank you so much, Dr. Smith, for a fantastic presentation. We have a few questions actually that are coming into the chat box now. So the first one is from Professor Ken Zaret, who's the director of Penn's Regenerative Medicine Institute. And Professor Zaret was asking how the engineering principles for making vascularized liver structures, how can that scale to what is needed for therapeutic application in humans?
That's a very good question. I have to shout out my colleagues at the Wyss Institute, particularly Jennifer Lewis's group, which has shown very amazing work of like really inducing vascularization with organoids. So they actually use organoids as a building block for tissue, and they actually create a large bath of organoids that actually can be used as like a slurry, where they can extrude vasculature within these large tissue structures. So I think with the advent of 3D printing and organoid technology, we can really get at this goal of introducing both really small capillary sizes, but also large vasculature, which we can perfuse, which is really important in their functionalities for sure.
Fantastic. Our next question is from Nicholas DiCaprio, who's a PhD student in Jason Burdick's lab. Nicholas asked, with your microfluidic device, when using collagen, did you ever run into collagen compaction issues? If so, how did you mitigate this?
Okay, good question. So that's a very challenging part of these microfluidic systems. We actually coat our microfluidic channels with glutaraldehyde that allows cross-linking of the collagen gels to the walls of the glass. Because when you use this needle extrusion method, you can actually pull on the needle and basically rip the collagen gel itself. And then also, when people use these systems for example, vasculogenesis assays, where you have endothelial cells within the channels and fibroblasts, people typically growth arrest the fibroblasts because they really contract on the collagen gels. So you can increase the collagen density to make the gels a little bit stiffer, but you can also use this glutaraldehyde technique, but you have to wash it out because it's toxic to cells. But that's one strategy that we use in creating these systems.
Great. We have another question from Beth Thompson, who's a big fan of shear stress, apparently. She said, thank you so much for the fascinating talk. There are so many potential clinical applications for this work, but what are some that you're most excited about? It seems very applicable to the study of biliary atresia, I think it's pronounced, but I'm curious what else you might be thinking about.
Oh, so this is really particular in terms of like the biliary tree. I think, so biliary atresia is a disease where you actually don't have the bile duct that's formed. So it's actually one of the largest demands for pediatric liver transplants. There's been pioneering work from Ludwig Valier's group. We're able to create like electrospun collagen scaffolds, and they can seed organoids into these tubular structures, and you actually can transplant these structures in vivo. So I think with some of these fabrication tools where we can create on-demand, scalable biliary tubes, we can begin to really think about ways where we can transplant these tissues. So there's a lot of room towards the liver space. And I think in terms of some of the cilia work, I think like polycystic kidney disease and polycystic liver disease, I think there's huge opportunity to look at the degree of ciliation in these biliary tubes and begin to understand those mechanisms and how they manifest into those types of diseases.
Great. We have another question as well from Hannah Horn. Have you ever considered using microscale 3D printing to generate your architectures, patterns, or microfluidic devices?
I have thought about it. I haven't fully utilized it, but a lot of people in Chris Chin's group and our group have used 3D printing as a way to rapidly manufacture these structures. I think with the needle pull-out method, of course, there are some technical challenges, but with 3D printing, you could just rapidly produce many different structures. You can print cells now. I think there's just like so much opportunity in the 3D printing space.
Great. Do we have any other questions from the audience? All right, I think if we don't have any other questions, let's thank Dr. Smith again for a fantastic first seminar to kick off the BE seminar series. Thank you so much again, Dr. Smith, for accepting our invitation to be our first speaker. And I think afterwards you'll be talking to some of our students. And to everyone else, have a great day.
Thank you.